Audition and balance. The ear is an organ that detects both sound and movements of the head. The funnel-shaped skin-covered external part of the ear is called the auricle and serves as a way to direct sound into the external acoustic meatus. There are fine hairs that guard the opening of our external acoustic meatus. Deep within this ear canal is wax-like secretions called cerumen, which mixes with dead skin cells that helps reduce infections. There's going to be a tympanic membrane, an eardrum, that vibrates when sound hits and provides a means for transmission of sound waves from the external ear to the middle ear. This is your middle ear, and this is your inner ear. The tympanic cavity of the middle ear contains three small bones, the malleus, the incus, and stapes. These are called the auditory ossicles, and they function to amplify and transmit the sound waves through the oval window of the inner ear. There are also skeletal muscles of the middle ear called the stapedius and tensor tympani that serve to protect the ear receptors by resisting any kind of abnormal amounts of vibration. There are two membrane-covered openings located in the bony regions that separate the middle ear from the inner ear. This is going to be the oval window and the round window. The oval window, which we just said, transmits sound waves to the cochlea, the inner ear, and the round window allows fluid in the cochlea to move and vibrate. Then the final part of the ear is the inner ear. This is located in the petrous part of the temporal bone, where there are cavities called the bony labyrinth. Within the bony labyrinth are these membranous, fluid-filled tubes and sacs that contain receptors for both hearing and balance. The bony labyrinth is structurally and functionally divided into three main parts. We have the cochlea, which contains hair cells and respond to sound and vibration, and it contains the cochlear duct. We have the vestibule that contains two membranous sacs called the utricle and saccule, which also have receptors for equilibrium. And then the semicircular canals, which are membranous semicircular ducts that also contain receptors for equilibrium, mainly detecting angular acceleration. And that's all inside the cochlea. Perception of sound. Sound is a perception of pressure waves that are established by vibrating objects and can move through any medium, transferring each particle's energy to the next particle. Pitch, on the other side, is described as whether or not the sound is high or low, and that depends on the frequency of the wave. The frequency of the wave is measured in a unit called hertz, which is the number of waves passing through a certain point at a given time. So inside the inner ear, there's a region called the spiral organ of the human ear that perceives sound frequencies between 20 hertz to 20,000 hertz. 20 to about 20,000 hertz. But the most sensitive region is going to be between 1,500 to about 4,000. High frequency sounds cause movement of hairs nearest to the oval window, whereas the lower frequency sounds move the basilar membrane that are near to the apex of the cochlea. So it depends on the location of the cochlea and which hair cells are bent. Then, of course, we have loudness or intensity of the sound depends on the amount of back and forth motion that vibrates the object. Soft sounds cause small movements inside the basilar membrane, whereas large sounds cause larger movements. And the loudness is then measured in a unit called decibels, with 0 decibels at the minimum, and then the highest on average is about 60 dB, 60 decibels. So the transmission of sound from the outer to the inner ear. So first and foremost, the cochlea in the inner ear is a snail-like shaped chamber that contains the spongy bone axis and a region called the modiolus. Two chambers of the cochlea are filled with a perilymph. This is a fluid called the scala vestibuli on the upper chamber, and then we also have the scala tympani on the lower chamber. So scala tympani will be below here, scala vestibuli will be up here. This vestibular membrane separates the scala vestibuli from the cochlear duct, and the basilar membrane separates the cochlear duct from the scala tympani. Both the scala vestibuli and scala tympani merge at the apex of the cochlea, creating something called the helicotrema, right in this region over here. This is called the helicotrema. The spiral organ, or the organ of Corti within the cochlear duct, is the sensory structure for hearing. Well, this is what you're seeing right here. This is the cochlear duct. The cochlear duct inside contains a fluid over here, shown in blue, called endolymph. And the spiral organ is thick sensory epithelium that consists of these hair cells and supporting cells that rest on top of the basal layer. So right here you have the basilar layer, and then you have the sterocilia of the hair cells right here. The two categories of hair cells rest on the basal membrane, a single row of these inner hair cells that function as a sensory receptor for hearing, and then we have 3 rows of outer hair cells that alter the response of the spiral organ to sound. The apical surface of each hair is covered by numerous stiff microvilli called the sterocilia, and there's one large hair that you can't see over here called the kinocilium. The stereocilia and the kinocilium are embedded in this very gelatinous structure called the tectoral membrane. So the sound waves are then captured by the outer ear, and they're sent into the inner ear in vibration via the tympanic membrane. The tympanic membrane begins vibrating and pushes the malleus, the incus, and stapes, the three ossicles that then vibrate the oval window. And then the oval window transmits these vibrations into the scala vestibuli, and as this fluid starts moving around, the basilar membrane begins to move. And depending on how these kinocilium and the stereocilium bend, that will then trigger a signal through the vestibulocochlear nerve to the brain. As we said before, frequency is measured in hertz, and this is the number of waves passing through a certain point at a given time. The spiral organ of the human ear perceives frequencies between 20 to 20,000 hertz, but it's most sensitive between the 1500 to 4000 hertz. So high frequency sounds produce the standing waves close to the oval window, and then low frequency sounds produce these waves closer to the apex of the cochlea. The apex is in the circle, and then the base is at the square region. The sound's transduction in hair cells. So the stereocilia of the inner hair cells are aligned with each stereocilium getting progressively taller and taller. The tips of each of these stereocilium contains an ion channel that is connected to the tip of the next adjacent stereocilium by this filamentous protein called a tip link. These tip links are what link them together from one hair cell to one hair cell. The first one is taller than the second one, which is taller than the third one. These tips are then bathed inside endolymph, which has a ton of potassium ions, giving the endolymph a potential of about positive 80 millivolts, compared to the cytosol of the hair cell being about 40 millivolts. So it's a lot higher charged. And as the motion of the endolymph is repeated, based on the basilar membrane moving around, the inner hair cells are pushed into the tectoral membrane, causing the stereocilium to tilt towards the taller neighbor. This then pulls that tip link and then opens up the ion channels in the shorter hair cells, allowing more potassium to move into the hair cells, causing depolarization. So as this bends, right here, you can see it bends over, causing the membrane over here to depolarize, ions rushing in, triggering a change in the membrane of this cell. Eventually, that's going to lead to cation flow into the cell. Calcium ion channels begin to open up, causing the vesicles of glutamate to be released. And the neurotransmitter glutamate is then released from the base of that inner hair cell and binds onto receptors of the primary neuron and creates these localized graded potentials that send signals towards the vestibulocochlear nerve. On the other side, when the tip links are slackened, or if the stereocilia, the smaller hair cells, bend away from the kinocilium, in the sense over here, the ion channels are closed. Therefore, there's no ions, there's no potassium or calcium entering into the cell. Therefore, there's no release of neurotransmitter. Therefore, there's no signal, and therefore, you do not hear anything going through the vestibulocochlear nerve. Equilibrium and head movement. The term equilibrium refers to our awareness and monitoring of head position. The sensory receptors inside the utricle, saccule, and semicervical canals monitor and notify us of our head position or equilibrium. So let's start with the angular acceleration first. At the base of each semicircular canal is a large area called the ampulla, and within the ampulla is an elevated region called the crista ampullaris. This entire structure right here is the crista ampullaris. On top of this region is an overlying gelatinous dome where the kinocilium and then the stereocilias are embedded in the cupola. The cupola is shown here. And you can see these tiny hair cells where there's one big one and several small ones on the right of it. One big one and several small on the right of it. So the crista ampullaris detects rotational movement of the head. When the head first rotates, the endolymph is going to have a slight lag behind the movement and pushes against the cupola, bending the stereocilias, which alters that neurotransmitter release and stimulates the signal to the sensory neuron. So you can see right here, as that fluid is moving from the left to the right, it's going to bend down the cupola in the opposite direction as of the head's movement. So if that head is rotating at a constant speed, eventually the endolymph catches up with the ampulla, so the stereocilias are no longer bent. And if these hair cells are no longer bent, there's no signal. When the head stops rotating and the endolymph continues to move, it bends the cupola and hair cells, therefore causing the neurotransmitter to release and signals firing to the brain. Now let's take a look at static equilibrium and linear acceleration. The sensory receptors found along the internal walls of both the utricle and saccule is the macula. The macula are composed of hair cells, again stereocilias and supporting cells, with each hair having one long kinocilium that is attached to the stereocilias and detect movement. The mass of the small carbonate crystals on top called the otoliths. These are little crystals. Covers over the gelatinous layer. So these otoliths are going to push on the underlying gelatinous layer, thereby creating weight of the otolithic membrane covering the hair cells. So when the head is held erect, not moving, this otolithic membrane applies pressure directly onto the hair cells, and the basal level stimulation occurs. If you tilt your head back or front, the otolithic shifts the position, bending the hair cells and changing the amount of neurotransmitters that are released, stimulating the vestibulocochlear nerve, cranial nerve number 8, to send signal. So just remember that bending the stereocilia toward the kinocilium causes the hair cells to depolarize, meaning the small ones bend on top of the big one, and the greater the frequency, the signals are generated. Bending the stereocilia away from the kinocilium, in which the small hair cells bend far from the kinocilium, the big one, it causes the cells to hyperpolarize, and therefore there's no release of neurotransmitter, and therefore low frequency signals. And the brain has to interpret the change in nerve signals and then determine the direction of the head that's being tilted. So remember, when the big hair cells bends down, this is going to be hyperpolarized, and then whenever the small stereocilia bend towards the kinocilium, this is going to be depolarized and send a signal to the brain. Auditory pathways of sound to the brain. Sensory axons of the cochlear nerve, primary neuron, terminate in the cochlear nucleus of the medulla oblongata, where they then synapse the secondary neurons to the inferior colliculus of the midbrain. So let's start from the vestibulocochlear nerve number 3. It works its way up towards the brain stem. It's going to hit a region called the superior olivary nucleus. This is where nerve signals localize for sound and monitor reflex for the sound that controls vibration of the ossicles. These nerve signals are going to then make their way into the inferior colliculus of the midbrain. And this is where it can monitor reflexes to loud sounds. This can alter the body's position to jump or even turn their head. And nerve signals are then transmitted to the medial geniculate nucleus of the thalamus for processing and filtering. Eventually, these nerve signals are transmitted to the primary auditory cortex inside the temporal lobe. And this is where the final processing and perception is conducted. Deafness is defined as hearing loss. Hearing loss can be defined as either conductive or sensorineural. Conductive deafness is when there's a problem with transmission of sound from the auricle to the tympanic membrane to the ossicles, whereas sensorineural deafness is a malformation or damage to the structures of the inner ear or the vestibulocochlear nerve.